your guide to genetics and genomics in the fertility clinic. The hashtag for this event is Pet Genetics IVF. This discussion has been made possible thanks to the kind support of the Scottish Government. I'm Sarah Norcross, and I'm the Director of PET, and I will be chairing tonight's discussion. When thinking about having a family, people are perhaps more inclined to think about genetics than at any other time. Indeed, who hasn't, had those sorts of conversations with people wondering about who the baby will look like when it is born? And then, of course, the experience of people looking at a new baby and saying, "Oh, he has your eyes and his dad's smile." Those are very normal conversations that people tend to have. Now, the development of assisted conception has been accompanied by far-reaching developments in genetics and genomics, and it can be challenging for patients, policymakers, and fertility professionals to keep pace with this technology. When genetics isn't your specialty, you might retain a grasp of the basics, but nonetheless struggle to make sense of the latest claims and approaches. Simply distinguishing between genuinely new technologies from earlier ideas that have been sort of repackaged, rebranded in new guises can be difficult, and can also involve navigating a sea of acronyms. We are thrilled this evening to have a panel of experts who will explore and explain the role of genetics and genomics in the fertility clinic. We'll be jargon busting, explaining the basics, as well as some of the latest breakthroughs. And the questions addressed by some of the panel will be, how and why might prospective parents and or their IVF embryos be screened or tested? Carrier screening has traditionally involved testing donors for certain conditions and testing some prospective parents to find out whether they carry specific conditions that have appeared in their family history. Some people have argued for routine screening of a greater number of conditions. So I think we need to think about whether such an approach is supported by evidence, and also whether it's practical and a cost-effective thing to do in an NHS setting. Pre-implantation genetic testing, PGT, has traditionally involved testing some IVF embryos for problems that could jeopardize a subsequent pregnancy and/or result in the birth of a child with a specific condition or predisposition. Some proposed approaches now look for a larger number of conditions or just traits in embryos. And again, I think that we need to think about whether these approaches are supported by evidence and whether they are practical and cost-effective in an NHS setting. So we have a panel of 4 who will each give short presentations. We have Dr. Jonathan Berg, Dr. Nicola Williams, Professor Zosia Miedzybrodzka, and Dr. Francesca Forzano.
So my job is to give you a very brief overview of basically genetic testing and inheritance in genetic disorders, hopefully giving you a little bit of background. So essentially, we're looking at genetic variation and how it causes disease. When we're dealing with monogenic diseases, where one genetic variant causes a single genetic disease, such as cystic fibrosis, then we have, well, this slide illustrates that you have about 30,000 genes. If we looked at the entire genome, there would be about 3 billion bases or letters in the genome, and we're looking for one genetic variation that causes a genetic disease. So we're looking for a single disease-causing spelling mistake in approximately 6,000 copies of War and Peace that is causing the disease you're looking at. So the main problem is that a genetic condition is caused by a single gene variant. in 3 billion other letters, which is the rest of your genome and all the instructions for making a human. And as an added complication, or to make it more fun, there are in fact usually around 3 million other genetic variants in the background that are the normal variation between people that don't matter. So I'm looking for a single genetic change in 3 billion bases in a noise, if you like, of 3 million other genetic variants. So this leads to the first really important point, is that when you're doing a genetic test, it really helps if you know where to look. I've given you pictures of 2 sequencing machines. We can sequence the entire genome, but we get a lot of data and we're looking for just one single change. The machines always appear to be grey and rather boring, and I would like them to be different colours to make them more exciting. But the exciting thing is you can sequence a whole genome for one individual relatively quickly, relatively cheaply. The difficulty is always analysing the data that comes out of it. To go further forward, we do need one other thing, which is family tree notation. So when you're looking at modes of inheritance, we need to draw out a pedigree or a family tree, and we do this in a standard format. And this will reappear in some future slides. And we have men as squares, women as circles, and then when they have a child together, there is a tie line, and then they could have a daughter or a son. And the other piece of notation is that I will colour in a symbol if that individual is affected by a genetic condition. So that allows us to draw a pedigree, noting who in the family is affected by the genetic condition. The first condition I wanted to look at, because it illustrates one really important situation, is achondroplasia. And this is a frequent cause of short stature with reduced limb growth, and it's frequently detected prenatally on scans with a characteristic growth pattern identified, particularly growth of limbs falling off between 20 and 22 weeks with previously normal growth and preserved head circumference. And I've seemed to have reiterated my family tree notation here. And you can see that frequently mum and dad are entirely normal, but you have a first affected child. Okay. And what has happened is that as the genes were passed on to the child, a mistake has crept into the genome that was not present in either parent. So this is a de novo genetic change that has caused achondroplasia in a child. And in this case, neither parent has the genetic variation. The genetic variation is new in the child. So what happens in the mind of geneticist is potentially quite an interesting question, but clinically I'm thinking, okay, everything in this pattern looks like achondroplasia, or here I've thought maybe hypochondroplasia, and I know that hypochondroplasia is frequently caused by de novo variants in this gene, so I can test that gene. So I know where I want to look, guided by clinical parameters. And when we're thinking about PGD or PGT, then de novo gene variants are an interesting question, because on the whole, they could be in any gene. So current practice in the UK would not be to sequence every embryo to identify de novo variants that could cause disease, but this is a potential future development. At the moment in the UK, we don't do it. We don't do it for good reasons, because it's complicated, it's difficult, and would frequently end up with data that we would struggle to understand. So we can look for de novo variants if we see something unusual during a pregnancy, a severe developmental malformation. And in that situation, if it's not chromosomal, we'd expect to identify a cause, perhaps in about 40% of previously undiagnosed severe developmental disorders. So we'd use it if we see something on a prenatal scan where there's no family history. But without a family history, it's not something we'd be considering for PGT. And the reason for that is every time we look for de novo variants, we find 60 to 120 in any individual. So even Prince George has 60 to 120 genetics variants that his parents don't. And as far as I know, none of these are causing a disease. So again, there's background noise, so we wouldn't know what we were looking for. So when we're doing PGT for a monogenic disorder, there has to be an identified genetic variant currently in the UK that we are looking for. And this is usually because we found it because of a family history of disease. So for example, we have autosomal dominant inheritance where you have 2 copies of every gene and 1 copy of the gene has a fault in it that causes an inherited disease. And this could be achondroplasia. In about half of cases, it runs in family as an autosomal dominant trait. In the other half of cases, it's a brand new thing in the family. Obviously, when you pass on the genes, it's 50-50. You pass on the faulty gene, the genetic variant, the child's affected. 50-50, you pass on the working copy of the gene, the child is not affected. And so in this situation, it is very straightforward to test for that genetic variant as PGT or prenatal diagnosis. And prenatal diagnosis could be invasive, or it could be now, in many situations, using non-invasive prenatal testing. The other thing when we're dealing with inherited autosomal dominant variants is we have to make sure the genetic variant we test for is predictive of disease. So in achondroplasia, if you have the variant, you have the condition. But there are some genetic conditions that are highly variable, and a genetic test is strangely not predictive of outcome in the pregnancy. We can look at autosomal recessive, and for example, here we have 2 German girls with cystic fibrosis, and this is caused by having 2 faulty copies of the gene, and you get 1 from each parent who are carriers. This is commoner when there's consanguineous parentage, but frequently there is no parental consanguinity. The situation is also complicated because we could test to see if an embryo is affected with CF, but actually new treatments for CF have completely changed the disorder really over the last 2 years from, well, we don't actually know how well it's going to improve outcome, but the current information and data is very, very promising that we are changing what CF is as a disease with precision therapy. X-linked recessive, when the condition travels on the X chromosome. So a boy who has an X chromosome with a fault on it and a Y will develop the condition. For example, Duchenne muscular dystrophy. So really, having had 10 minutes, I think I'm almost there. And my final message is the most important thing. We have fantastic tests. We can sequence an entire genome, but we generate a lot of data. And the best way of coping with that data is to always have a good idea of what you are testing for before you arrange a test. And that is really the current situation in routine clinical practice in the UK.
We will move on now to the next speaker this evening, and that is Dr. Nicola Williams.
Firstly, just to say in terms of this presentation, the vast majority of the images and pictures that are in it have been supplied by either the PGT laboratory team in NHS Greater Glasgow and Clyde or NHS Lothian. So thank you for that. So pre-implantation genetic testing involves genetic testing of embryos prior to implantation. And it, depending on what type of genetic abnormality you want to look for in a family will depend on what type of genetic test you need to use. And the other thing just to say is certainly based on what we're currently doing in Scotland, although not necessarily where we would like to get to in Scotland, depending on what test you use will also depend when you need to biopsy your embryo. Apologies for all the embryologists and co on the call because this is not news to you, but just a quick run through of embryogenesis days 0 to 6. So we have fertilization at day 0. And by day 3, we have a cleavage stage embryo where we have roughly 6 to 8 cells in the embryo. And for some of the tests that we would do, particularly in Scotland, we would do a biopsy of between 1 and 2 blastomere cells at this stage. And by day 5, we have progressed to a blastocyst, where we have an inner cell mast and a trifectoderm outer cell ring. So at this stage, if we were doing other genetic tests, we would do a day 5 blastocyst biopsy where we're taking multiple trifectoderm cells. And so it's very dependent on what we need to do and when we need to do it. Again, no images that are very familiar to most of the people on this call. So a day 3 blastomere biopsy shown here on the left. You can see on the left of the first picture, you have the pipette actually holding the embryo in position, and a tiny, tiny little hole is put on the side of the embryo, and we remove 1 or maximum of 2 cells from that. And you can see that on the right hand side, with very fine needle put into the embryo. So this is a removal of single cell cleavage cells removed, and I think it's fair to say that we know that this results in a lower pregnancy rate, and that's for a variety of reasons, partly because some of these embryos won't progress to a day 5 blastocyst stage, and perhaps because there's some evidence that doing the biopsy at this stage may compromise the embryo itself. And then the second type of biopsy that we do in Scotland is a day 5 blastocyst biopsy. So again, you see the pipette on the left-hand side holding the embryo in place, and the fine needle on the right actually taking some of those trifectoderm cells out. And again, we know that there's a higher pregnancy rate associated with a day 5 blastocyst biopsy. So ideally, this is where we would like to get to in Scotland. We'd like to move away from our day 3 blastomere biopsies onto our day 5 blastocyst biopsies, but we aren't able to do that at the moment. So just moving into the genomic side of things. So I'm going to talk to you first about PGT-SR and chromosomal abnormalities. So this is a normal human karyotype. And really just to say that a normal human karyotype will have 23 pairs of chromosomes. So you can see the chromosomes here labeled 1 to 22, and then we have X and Y as the 23rd pair. And we inherit 1 chromosome from our mother and 1 chromosome from our father. And the X and Y shows us that this particular karyotype is male. The example I'm going to talk to you about next, we're going to look at a chromosomal abnormality for chromosome 6 and chromosome 1. So this is an ideogram of what these chromosomes look like, so more of a cartoon diagram, and you'll see the banding patterns for each of these chromosomes. So here we see 2 normal chromosome 1s and 2 normal chromosome 6s. So you can see that the chromosome that's inherited from mum and from dad appear normal. And this is what a structurally rearranged chromosome would look like. On the diagram here, you'll see we have 1, I'm hoping people can see my pointer, but you'll see we will have 1 normal chromosome on the left, and on the right, this chromosome 1 has a little bit of this chromosome 6 stuck to the bottom. And here we have a normal chromosome 6, and this chromosome 6 has the remainder of this chromosome 1 stuck to the bottom. Now, this is a balanced translocation. So in a family where there's structural chromosomal abnormalities, the parent will carry this structural abnormality, but will not necessarily have any disorder at all, and it's in the balanced form. It's only when it's inherited in the unbalanced form in the embryo or child that you see the genetic disorder. So how do we do PGT-SR in Scotland? So we do still use a FISH assay. So this is fluorescent in situ hybridization. And how that test works, you'll see here we have coloured blobs which will stick to the chromosome. And these are DNA probes which are labelled with a fluorescent tag, which fluoresces when we look at them down a microscope. So what we're able to do is put fluorescent FISH probes on at the ends of chromosome 1 and chromosome 6, and then we can also put telomere probes here as well, centromere probes here as well, so that we can actually look for patterns for these chromosomes. And this is what fluorescent in situ hybridization test looks like for a single embryonic cell biopsy. So the first cell I'm showing here is the maternal cell, and you will see this is normal. So if we go back to this previous picture, you will remember that there are 2 chromosomes, both in the balanced and the normal form, where we have probes that will stick to the bottom of chromosome 1. We have the same at chromosome 6, and we have these centromere probes. And actually, when we look at the maternal cell, you can see we have 2 greens, we have 2 reds, and we have 2 blues. So totally normal. This is an embryo biopsied cell, so a single cell with FISH probes applied. So again, we can see 2 green probes, we can see 2 blue probes, and we can see 2 reds. So this is normal. Now, we are not able to tell with this test if this is in the balanced or unbalanced form for this embryo, but the embryo doesn't have an unbalanced structural abnormality. And then for chromosome embryo number 2 here, we can see we have 2 blue probes, we have 2 green probes, and we have 3 red. So this tells us that embryo 2 is abnormal and has an unbalanced structural abnormality. So just moving on to PGTM and single gene disorders. So single gene disorders are tested differently than what we would do for a structural abnormality. So it requires a different genetic test. And there's a variety of ways of doing that. And I'm not going to really go into much detail about them today, but we can use microsatellite markers, we can use microarrays, and we can use next generation sequencing. I think it's fair to say most people have moved or are moving towards next generation sequencing for their PGTM services. So PGTM can be used to detect, I'm just going to give an example of an autosomal dominant condition such as Huntington's disease, but they can actually be used to detect other chromosomal modes of inherited disorders, so autosomal recessive, etc. as well. For the vast majority of PGTM, we don't actually, although we know the genetic mutation in the family, we don't go looking for the genetic mutation itself. Instead, we want to identify the high-risk chromosome. So this is a family with an autosomal dominant condition here, and you can see we have got the maternal grandmother is affected, and the mother is also affected with the autosomal dominant condition. Now, what we're able to do is apply a genetic test that allows us to identify the chromosomes in these families. So we have a dark grey and a light grey from the maternal grandfather, and for the maternal grandmother, we can see that we have a dark blue and a light blue. What you're then able to do is track these chromosomes through the family. So when we do the genetic test, we are able to see down here that this embryo is normal. It's inherited the light orange from dad, and it's inherited the light grey from mum. So it's inherited this chromosome from the grandfather. But what you can see here is in these 2 embryos, actually, we've inherited the dark blue chromosome from the grandmother and the mother. So these 2 embryos would be affected. So what we would have to do when we're looking at these families is we would do a microsatellite test, and we would look to track the chromosomes within the family, and we would then use that to identify the high-risk chromosome that we know contains the mutation in this family. So this is just a trace of some microsatellite markers just to show you what this looks like. Again, I'm not really going to go into this in much more detail other than to say this is a PCR-based test which makes lots of copies of the chromosome patterns at a specific area on the genome. So this would be the chromosome that contains the genome of interest. So for Huntington's disease, that would be chromosome 4. And what you can see here is in all of these traces that we have 2 high peaks, and they are kind of highlighted by these grey sections. So this high peak here would be one of the chromosome patterns from one of the grandparents and the other one from the other grandparents. So we have that for mum. You can see that dad's pattern looks different. And then we have 2 embryos down here. So we've got a maternal sample at the top, father at here, embryo 1 and embryo 2. Now, when we actually look at tracking those chromosomes, we can actually see that this chromosome from this microsatellite marker has been inherited by embryo 1, and actually embryo 2 also has the exact same chromosome. So if this is the high risk chromosome from mum, we are able to tell by this test that the embryo has inherited the high risk chromosome. And then when we look at the paternal chromosome, we can see this is the chromosome that's been inherited from dad by the embryo. So we're able to tell that these embryos are affected with the disorder without doing the genetic test as well, just by tracking the chromosomes. So this is a set of results from a PGTM output for a family. So this is a family that have gone through around of BGT, and we've had 6 embryos in this family. And here you can see the pink chromosome from mum, which is the affected chromosome, and the blue is the normal. We have embryos 1, 2, and 3 who have inherited the pink chromosome from mum, so they are high risk and would be predicted to have the disorder. And we have 3 embryos here who have inherited the blue chromosome from mum, so would be low risk and are predicted not to have the disorder.
Our third speaker this evening is Professor Zosia Miedzybrodzka. She is the Professor of Medical Genetics and Director of the Centre for Genome-Enabled Biology and Medicine at the University of Aberdeen. She is the Clinical Service Director of Genetics at NHS Grampian.
So I'm hoping that as I talk, I will be building on the conversation we've had earlier from Jonathan and Nicola, and I hope I take you all with me. If you have queries about things I'm talking about, obviously we'll pick that up later. Carrier screening, now I'm going to start talking about just carrier screening in general, is where in healthcare we think about testing for people who've got a carrier of a genetic condition, as in they themselves are unaffected by the condition, but they're at risk of passing it on. That's typically what we mean. And this is usually the case in those patterns of inheritance Jonathan mentioned earlier, the autosomal recessive and the X-linked. And carrier screening can be offered either antenatally, which is actually where a lot of the trials were done in the 1980s and 1990s, or preconception. And preconception is more of the arena we're probably talking about here now. If you find what you're after here is to find a couple at risk of having a child with a particular disorder that they're not alerted to by their family history. And the options therefore are prenatal diagnosis, PGD, as we've heard, but there would also, at least in theory, be the option of choosing gamete donation, or just simply limiting family size, or I think I've done donors twice, you know, we often see folk going down the prenatal diagnosis, and the PGD route is increasingly common, but limited by supply and limited by the personal costs of going through the treatments, which can maybe be discussed later. But in healthcare, I think the decisions as to whether to do carrier screening have been determined by a combination of clinical utility, as in how it changes management, and economics. And typically in our populations, we in the UK, there are very limited groups really where carrier screening is in place for single conditions. So what's available right now in the UK is historically driven partly by ethnicity, and fertility donors get a bit more because they have more children, particularly sort of thinking about sperm donors, but also egg donors. So in the UK, the carrier screening available generally is for sickle cell disease and thalassemia. And those are largely not done using genetic tests as the first line. They're done by looking at, so it's a very cheap way of looking at the full blood count samples that come into hematology and getting an extra test done on them. And then in selected populations, for example, people of Jewish ancestry have the option of asking for Tay-Sachs testing, which is Tay-Sachs is a very rare disorder in the rest of the population that causes, through early childhood, neurological deterioration, and usually demise by around the age of 4 or so. So these are conditions that are sort of certainly the sickle cell and the thalassemia testing is widespread across the UK, whereas I would say the uptake of Tay-Sachs screening is very patchy, and it's certainly, I think, more available in populations where there is a higher number of Jewish people. And that's traditionally at least done using biochemistry. But there are options now to move to genetic ways of testing for these things. But basically the stuff that's in place is really, pretty cheap and done where the carrier frequency is highest. So for example, there was decisions made that for sickle cell and thalassemia, that they were common enough that we could do screening in the whole population there. But that is currently only offered once you're actually pregnant and babies are tested after birth for sickle cell. So it means that you can't, it's sort of not available if you're just thinking about it unless you ask. And typically, if you were to ask beforehand, it would be people who have a higher risk due to their ethnicity that would be offered that testing. And typically it's Mediterranean and sort of sub-equatorial African origins that are the triggers. So always talk about in Aberdeen, which is sort of the furthest north genetic centre in the UK, and you would think that we have a very low number of people from of non-Scottish origin in the whole of the region, but actually in Aberdeen city we have a very large Nigerian population driven by the oil industry. So we have one of the highest rates of testing for sickle cell disease, so which is a bit of a paradox. So moving on, thinking about fertility donors, there are some extra tests offered, typically looking for chromosomal rearrangements and also for cystic fibrosis. Cystic fibrosis carrier status, with 1 in 25 of the Caucasian population being carriers of cystic fibrosis. So that's that chesty condition that Jonathan spoke about earlier. So this slide is just to remind you all, as Jonathan said, that not all conditions can be screened for. So if you're looking to make sure your child doesn't have anything wrong with them, if you were to do a test that did more recessive conditions and not just those few that are not, only available in limited circumstances, if you sort of said, well, they're not worth doing individually, which was the sort of historic decision, but actually, what if we just tested for everything all at once? That's the premise of extended carrier testing. Well, what would we miss? And yes, you would get, maybe you could perhaps aspire to picking up autosomal recessive and X-linked conditions, but you would not be able to screen preemptively to detect the de novo conditions, as Jonathan described earlier, for example, those autosomal dominant conditions, which actually form a really large proportion of children born with learning disability, at least those that we can test for. And obviously, it doesn't tell us anything about chromosome diseases like Down syndrome, which again are typically not inherited, and any disorder that happens as a result of the environment or post-zygotic disease. So the premise of extended carrier testing is to look at, say, 400 or more genes, or everyone as many as you can think of, to look for couples where they are either both a carrier or a carrier of an X-linked condition, with the aim that you could offer a prenatal diagnosis or PGD. And typically, if you do it, the more genes you look at, the more abnormalities you might find. So Jonathan was reminding us about how, something like 1 in 1,000 bases of our DNA are different. And if we look at how many disorders we're carriers of, then basically, if you look across the whole genome, we're all carriers of quite a few things. So it only matters if both parents are a carrier of the same thing. So the more genes you look at, the more you end up having to test both parents, which of course doubles the cost of your screening. And if parents wouldn't change what their reproductive intentions as a result of the test, you might argue you shouldn't be doing it. And you know, the limiting factor really in doing this type of testing, I'm going to speak about a little bit more length, is just the practicalities of being able to achieve that perfection of picking up every rare condition, every recessive condition. Now, so one way around that is to test the couple, but it is sort of straightforward if you focus on common variants that are well known to cause disease. But if you're looking for a common recessive variant, there are a few that are common, but many that are rare. The problem is if you've got a common one in one parent, you kind of, to be sure the other one won't get it, you've really got to check for the very rare things as well. And that's where the limiting factors are. Another thing to think about is, well, what are you actually going to screen for? Should it be childhood onset conditions only, or adult onset conditions? What do you call serious enough? What can we screen for? And what if the donor or the person you're testing or the person wanting to know has a disease they didn't even know about, and you find that out as a result of the test? Perhaps rarer if you're doing targeted testing, but all things you have to think about. And this is just an example of a paper where they implemented carrier screening using high throughput next generation sequencing. So there could be two approaches. One would be you just look for common variants that are more common in the rest of the populations, and that's what's listed here. And if you just tested for them, you could be pretty sure, you know, what you might be looking for, and therefore be sure that those changes would cause disease. But the problem would be whether there's rare variants there on it, perhaps in the other parent that you missed. But you can see from this how the frequency of variants can vary from anything between sort of 1 in 30 in some populations with cystic fibrosis, it's commoner in Scotland, to, you know, 1 in 700, 1 in 2000, and so on. So you're testing a lot of negatives to find positives for each gene. But if you collectively lump everyone together, then you are going to find quite a lot. That particular gene has 6,128 letters in its DNA code, and there's over 1,700 mutations being proved to be pathogenic, and only 90% sort of hit the top 30. So would you test everyone for all of those variants? And then if you find, you know, where you've got a common disease like cystic fibrosis, you can be pretty sure what's pathogenic, but it's a lot of searching through. But if you are testing rarer diseases, having a list of what sort of the truth set of variation can be really, really difficult. We have looked in the past in Scotland about the idea of maybe doing a carrier screen for the commonest variants, but the problem is you might miss so much. And where it's particularly difficult, where you find variation that you can't prove. And I was hunting about in the literature to just find some examples, and it's interesting that there's most of the evaluations are really around people just saying they've offered a service. And most of the literature comes from commercially driven testing. So one particular test I found, just, you know, with a simple Google, 625 Canadian dollars. I presume that's per parent. So you have to double that for 2 parents. And looking at that type, the evaluation of that, 70% of the singletons tested positive for something, and allegedly 1 in 40 of the couples were said to be both positive as a couple. But then what action did they take? How much actually validated? And this paper here gave a number of examples of carrier testing rates from various commercial platforms reported to a center where they'd gone on and evaluated what those differences were, and whether, so this had been sort of highly automated at scale analysis, and actually there was a lot of changes that had been reported as pathogenic that actually weren't. For example, there's a case there where a child was, a parent was thought to be a carrier of Duchenne muscular dystrophy, an X-linked condition, and they tested a fetus, and the fetus had it, and then they went on and tested the unaffected brothers of that fetus, fortunately, I think before the termination, and actually found that both the brothers shared the variant, just illustrating how important it is that you know what you're dealing with. If you try to put this type of thing in, you're going to have a lot of resource. You're going to have a lot of scientists who can look at the variants and validate them. You've got clinicians to explain it. And really, although it's something we could do, right now it's just not been top of the list of priorities because that complexity of doing it really, really well. In Scotland, our biggest priority right now is actually to do more testing for testing people's cancers to see what drugs they should be on. And we're still wanting to improve our services for diagnosing people with known conditions, where we're thinking about planning, they might be thinking about planning the next pregnancy. So it's about priorities. So yes, you could do it, but there's lots of limitations. It uses a lot of resource, and the current commercial platforms are limited. Thank you.
So our final speaker this evening is Dr. Francesca Forzano. Francesca is a consultant in clinical genetics at Guy's Hospital and honorary senior lecturer at King's College London. She is chair of the Public and Professional Policy Committee of the European Society of Human Genetics and lead author of this committee's statement on the use of polygenic scores in pre-implantation genetic testing.
So I am going to introduce yet another consonant this evening. So we are talking now about PGTP after having talked about PGTM and PGTSR. PGTP stands for pre-implantation genetic testing in the form of polygenic risk score. So we have to consider here two elements. One is the context. We are discussing about doing a test in the context of pre-implantation genetic testing. And I will not concentrate much on the context because the speakers before me have discussed already a bit about this, but just to highlight that we are considering a situation where you have to conceive your embryos in vitro in order to have a test feasible before the implantation. And this is applicable to couples who are infertile, but might also be applicable to couples who are fertile, but they would like to have a test in their embryos before implantation because they might be at risk of a condition and they do not want to have children affected. The tool we are discussing at this point in time is called polygenic risk scores. And the scientists who are currently doing research in this area are now actually changing slightly this name, and they tend to call them now polygenic scores instead of polygenic risk scores, precisely because there might also be some doubts regarding their actual effectiveness in assessing a risk. So I will discuss mostly regarding the polygenic risk scores tonight and whether we think they are meaningful when applied to PGT. So what are PRSs, polygenic risk scores? So first of all, it is not a test aimed to detect one single genetic variant, but it is a group of genetic variants which are per se very common in the population. And they have been selected in a certain way because it seems that this match is more common in adults affected with a given condition. And this is usually a common disease or a common trait. So we are moving from rare disorder, genetically determined, to common disorders where the likelihood of being affected comes from a variable combination of a number of the environmental factors, lifestyle choices, and some genetic predisposition. And the weight of each of these three groups might be quite variable, not only in each different common condition, but also from one individual to the other within that specific condition. So to create a polygenic risk scores, we collect hundreds or even thousands of variants all together. They have to be tested, detected, and then we combine them. And this, even nowadays, where we have been able to create quite a few of these for a number of conditions, do only capture a part of the genetic component of disorder. It's not the whole story. This is still an area under development. All these variants that we detect in our DNA can be located within genes, but also outside genes. So the vast majority of these variants, we have no clue on what they actually do. This is an association. So we find the variant ABC in the individuals with this condition, but we have no clue on whether the variant A, B, or C does something in relation to the expression of some genes and their function within the cells. So this is vastly, at the moment, a research domain. There are some pilots for some of the conditions coming along. One is within the UK for a cardiovascular condition. This is called the HEART project, but this is still to understand whether the application of some of these in specific contexts might actually be helpful in order to prioritize, for instance, treatment or some screening at the second level. It's important to highlight that the polygenic risk score per se does not exist, but it is constructed by scientists. So this is a main difference comparing to rare disorders. So for instance, at the beginning of the talks, we had an example of achondroplasia or hypochondroplasia. This is due to a discrete single nucleotide mutation that occurred when that individual was conceived, and it is like discovered. So I'm going to look into that gene and I check whether that variant is present or not. In this case, I do look into the whole DNA, in the whole genome of that individual. I look for a number of potential variants. I can variably select some or others and combine them in a different way and use different software pipelines in order to create my score. Okay, and also the way these have been created, is in a way artificial. So we have selected some of these variants on the basis of a specific research question. Okay, and the way we have made our questions within the research project and define our method influence which kind of variant we can identify as associated. So if you change something in the track, you might also end up to have different elements as output. So what are currently the limits for the PRSs when you consider the possible application to clinical settings overall, not necessarily in the context of prenatal diagnosis or preimplantation genetic testing, but in the context, let's say, for which they have been created and originated, which is a context of adults affected with common disorder. So first of all, the PRSs do not define an absolute risk, but a relative risk. So the absolute risk is usually only slightly modified. So to give you an example, in the general population, there is a risk of about 1 in 100 to develop schizophrenia. This is usually age dependent because it's more common in the young adulthood to develop this condition, but this is the risk for the general population. Okay. If you analyze the PRSs for a group of young adults, you might identify a subset who, according to what you have identified as a genetic predisposition, have half the risk of the others to develop this condition. So this polygenic risk score is significant because it's 50% less, so it's quite a lot, but the absolute risk of these individuals will be 0.5% to develop this condition instead of 1%. So the absolute risk is not the same as the relative risk. And this obviously has a huge impact in the clinical utility when applied to a clinical setting. There is the problem of pleiotropy. So we discussed the fact that you have to combine hundreds or thousands of variants, and many of these variants might also have some effect on other polygenic score for other conditions. And maybe they go in the same way, so they might confer a higher risk or a lower risk, but they can also go in the opposite way. So they might increase the risk for something and decrease risk for something else. We are currently constructing some of these PRSs for a discrete number of common disorders, but there are many, many others for which we still have no clue. Everyone who is involved in the research of PRSs say that these are most useful to use if you combine this together with the age of the individual, the environment to which this individual is exposed, and also the lifestyle choices. If you take out these elements, the actual utility of these PRSs is probably not that much because it is completely out of context. And also remember that they were actually analyzed and defined initially on an adult population where at least some information on lifestyle choices and environment were already available. So you also have already the information of the exposure of that individual to a defined environment. There might also a lot of confounding factors. So for instance, if that individual has also a proper mutation, let's say in a gene that will confer an autosomal dominant risk for something, obviously this will be more important as genetic component, but maybe we do not know that individual has a risk which is determined by something else because we have not looked into that, because this is not part of the PRSs that we have analyzed. And maybe because there is no family history, we could not incorporate this in our information because maybe this is a de novo change. So for instance, this individual might have a de novo change in the BRCA1 gene, having a high risk to develop breast cancer. But actually, if it is a de novo mutation in the family and we construct the PRSs for that individual and many others, this will not be an information that I use. And obviously, this will have a much higher weight than the PRS score in that individual. Okay. Another limitation is that the vast majority of these researchers use individual of European descent. So we have very little information about individual of other descents, and this has a huge implication. You might think that one mutation has the same effect everywhere in the world. This might be true for some rare disorders. So achondroplasia is always invariably associated with a specific mutation in FGFR3, whatever is the sex of individual, the background of the individual, they will always have exactly the same condition. For PRSs, this is not true. So we know that some PRSs might be associated with a condition positively or negatively in Europeans, but this association is not true in another group of people with a different descent. We don't know why. It might be for a complexity of the genome, for a linkage disequilibrium, or maybe for environmental exposures. So there are a number of factors that we still don't know, but the issue is that we don't have even enough population of other descent available for research in order to implement our knowledge in this field. This is a limitation in the validation. And also, as I mentioned, we can construct different PRS scores even for the same condition. And there is evidence that if you apply different PRS scores to the same population, you might end up with different risk estimates for each individual. So there is no reproducibility of different PRSs for the same condition. So this is obviously a warning because at the moment, there is no PRS score who has a definite clinical utility demonstrated in order to be applied in clinic. So we are starting to apply some of this, but they are still in a pilot phase. So we do not actually know whether using them will actually improve significantly our outcome in terms of healthcare. Okay, and this is in their original context, adult population with a risk as anyone has of a common disorder. Okay. If we consider a recent recommendation of the International Common Disease Alliance, so basically it is the association of researchers who analyze polygenic risk scores. They identify them, they publish on them, is their area. They also highlighted these problems, and they have launched like a warning that these are not valid on people of no-European ancestry. And also the information to be provided to the population should be very, very attentive and only be delivered by trained professionals because they can be very easily misinterpreted, which can mislead completely also their care if they're not used in an appropriate way and prematurely implemented. Also, there is a risk to exacerbate inequities in healthcare because, as we have highlighted, these can be maybe used for European ancestries, but not for all the other ancestries. And what to do with people with combined mixed ancestries? This is still obviously an area of research. So, taking this tool and applying this tool in our context, which is pre-implantation genetic testing, is yet another can of worms. So at the moment, we are not using PRSs routine in clinic in the context for which they are researched and studied for, but some private laboratory, particularly in the US, have started selling this test within the PGT genetic test market. So these are offered to embryos as a screening tool. This is not a diagnostic tool. It is a screening tool, according to them, to select the embryos or to rank the embryos with a higher or lower chance of a common disorder in future. Okay. So this is a screenshot for one of the websites. This is genome prediction. As you can see here, you can select your condition, in this case, type 1 diabetes, and your ancestry. Well, actually, you can only select European ancestry because the other ancestries are not really validated. And here you can see, you can select whether there is no known family history in that family or whether there is a family history, because obviously the genetic component in this case can be higher. If you can see here, this is obviously the best available score that they might achieve, which is a reduction of 29% of the condition or 46% of the condition. But this is again a relative risk. If you look at the absolute risk, if you don't have a family history, the risk goes from 0.4 to 0.28. So you have to make a rank on the basis of that score. If you do have family history, it goes from 5.5 to 2.9. But this is again allegedly, because we do not know whether this is the right population to test. We have no information about the context. We have no information about the future lifestyle choices. We have no information about the environment in which this kid will grow up. And with the climate change, will definitely be something quite different to what we know now. If you want to score for actually more conditions in a row, well, it's your lucky day. As you can see, these are the maximum risk reduction that you can achieve for each of these conditions. So what you can have is basically a figure like that on which you have to make your selection. So obviously there are added limits when it's applied to embryos, in that this is not validated in this population. We do not know whether this variant might have any effect in embryonic development. Within one family, you might have a small number of embryos to test with a very limited diversity between siblings that you can have in a very large population. And as we have seen, the embryos will develop mitotic mutations. So there will be de novo mutation that you can't test for. And all the embryos will be at risk for something. And there is also a risk that we will move from a clear-cut condition, a disease, which has a significant effect on that individual, or to specific trait. So for instance, to autism, or for other traits that for individuals are not a disease, but also part of their identity. So for this reason, and because of this limitation and all the ethical implication, the European Society of Human Genetics has released this statement saying that at this moment in time, the polygenic risk score applied to pre-implantation genetic testing at best is premature. ESHRE, the European Society of Human Reproduction and Embryology, has supported this statement. The American College of Medical Genetics has also released a similar statement. This tool is not currently considered appropriate when applied in pre-implantation genetic testing, but I will leave you with actually a question for you. What is our clinical question here? What is PGT aimed for? What our society wants to analyze in this context and to offer to our patient, which are the couple and the parents, but also to our future children.
It's important to note that in England, no one is doing day 3 biopsy or FISH testing anymore, and that it's not what would be offered to patients in England as better methods are now available. So I think that's perhaps one for you, Nicola, because it came from your presentation. So if you would like to address that, please.
Yes, no, that's fine. Thanks, Sarah. So yeah, I think ideally we would like to move away from day 3 biopsies, and there are a couple of factors that have influenced why we haven't been able to do that. So the Glasgow laboratory actually validated the Agilent 1 PGT assay, which is an NGS based assay, and the plan was to move our PGT SR service over to that. And just as we were about to move into our second phase of validation, the company pulled the kit from the market. So that has been slightly challenging. In Scotland in general, our genomic services don't have a bioinformatics team, and that's something that we are working on as part of our genomic strategy. So we're in a different position from the GLH models in the NHS in England, where they have a bioinformatics team. So to actually move our PGT SR service to microarrays or NGS would be incredibly challenging without having a kit that actually comes all singing and dancing and includes the analysis component, which is what the 1 PGT assay did with Agilent. We would have been able to analyze that data without that bioinformatics team, and we would have moved across. So it is slightly frustrating. What we are doing though is we are working with Guy's, the PGT lab at Guy's, and we hope to be able to offer Guys are planning to work with us to hope to be able to offer PGT-SR for Scottish patients. So we would move to that day 5 biopsy at that stage. I think it's important to say though, there's another question about suboptimal testing. So yes, there's evidence it's better to do a day 5 biopsy, and we know that, but I think this is a recognised diagnostic test and is still of good enough quality. GenQA do deliver external quality assessment scheme for PGT by FISH on day 3 biopsies, and although we are the only NHS lab in the UK doing that, and remember there aren't many NHS labs in the UK doing PGT, there are a lot of private labs, and the funding is a different issue for them, but actually there is an EQA that is run every year, including next year, for these services, and there are still a significant number of European labs who do this. So I think it's really important that we don't badge this as not being suitable for diagnostics, because it is. We would like to move to day 5 biopsies, but we're just challenged around some of our infrastructure at the moment.
Thanks very much for answering that, Nicola, and also for dealing with Karen Doy's question there as part of the same package. So I hope that's satisfied Karen as well. So the next question is more of a general question, really. And so I think all the panel may want to chip in, I don't know. But an anonymous attendee has asked, do we know whether IVF itself has adverse genetic or epigenetic consequences for children who are conceived in this way? For example, does the embryo culture medium make a difference? So are there any concerns about that the panel know about or would like to hypothesize on?
What it is known is that children born after assisted reproductive technologies overall have a slightly increased risk of congenital anomalies comparing to the children born in the general population. So on average, we give a risk of about 3%. for the general population, and we give a risk of about 4 to 5% to children born after assisted reproductive technology. How this is composed is a bit more problematic. So for what concern what causes it, there is no definite evidence at present. So some studies will suggest that it might be at least partially be due to the reason for infertility that in many cases not obvious or clarified of the parents. In other cases, it will seem that it is more attributable to the technology per se, which could be a combination of the ICSI, of the fact that the, not just the medium, but the whole environment in which the embryo is conceived and kept for a few days is different than the physiological one. There is currently a very, very large study ongoing with big numbers that aims to provide an answer to that. They have anticipated some of the results recently at one of the American conferences of human genetics, which will suggest that possibly is a combination of factors. So each of these components within the technical process, but also the infertility might have a contribution. For what concern which kind of conditions, this is a very important question. At the moment, there is only clear evidence that there is an increased risk for some condition with an epigenetic effect. So for instance, for Beckwith-Wiedemann syndrome, where the risk in the general population will be about 1 in 10,000, the risk seems to be about more than 4,000. Which is a lot more, but again, in terms of absolute risk, is still a rare disorder, is still rare. There is no current evidence that there is increased risk of autosomal recessive disorder, which is linked basically to what Zosia was saying before. So, albeit the expanded carrier testing is aimed to identify individuals with recessive condition, there is no evidence that people undergoing assisted reproductive technology because of infertility have an increased risk of recessive condition. So that would be a completely separate package for their treatment that will not seem to be related to an evidence of benefit in relation to their infertility. But this is so far what we have in terms of clear-cut evidence. But obviously big casuistry are still under study in order to be more, to have more details and to understand whether we will be able in the future to tailor a little bit also our counseling and provide a more personalized risk, let's say, to our patient.
A paper in Lancet Digital Health earlier this month proposes a non-invasive way of detecting embryo aneuploidy using morphology, morphokinetics, and algorithms. Aside from therefore avoiding the need to biopsy the embryo, would this be more useful than PGT-A, or would the same criticisms that are applied to PGT-A still apply?
PGT-A is not supported in NHS Scotland, and I think it's not supported in NHS England as a diagnostic test because of the differing opinions around it. So we wouldn't do it using a genetic test.
Does anybody else want to come in on that? I can see you nodding, Francesca, in confirmation that it's not available in England on the NHS. Is that correct?
Yes, and I will also say that the same criticism will apply.
Do you want, for the purposes of the audience who might not be familiar with the criticisms of PGT-A, would you like to just sort of slightly expand on that, please?
Yes, so PGT-A is basically the analysis of aneuploidies. So it aims to detect the number of chromosomes of that embryo, but also applies to not necessarily just the number of chromosomes, but potential big imbalances that might impact, obviously, the viability of the embryo. Many, mostly private labs have done a lot of studies because they are offering this routinely. So they have obviously the largest casuistries, but even recent meta-analysis, there is a big work, it is a Cochrane review, which was published in 2020, did not find any evidence of clinical utility in offering this extra screening. This was originated many years ago with the consideration that if we will be able to check whether the embryo that we are going to implant might have, for instance, trisomy 21 Down syndrome, and we can avoid to select this, we will have a better chance to have a pregnancy. So the only result so far is that this technology at this moment in time is probably helpful to reduce the failure of implantation, but there are no differences in the chance to having a healthy live-born child at the end of the day. So is higher expense, and there is also a higher chance also to damage the embryo because you will do these extra tests and so forth. So there are a number of technical and biological doubts behind this. There is definitely some more research and implementation of it. So obviously in the future we will see, but at the moment there is no evidence that it is actually effective.
Thanks very much, Francesca. So the next question, or it's a comment really, is from Sarah Bennett Day. And she says, thank you to the third speaker, so Zosia, for highlighting the impact on clinical practice and high levels of resource required for extended carrier screening. In antenatal screening, we're seeing a huge impact from the relatively recent introduction of rapid exome sequencing, an increase in both interruptions of pregnancy and on requirements to provide earlier testing in future pregnancies and prenatal testing. And I think that then is a sort of really important point, I think, particularly in the current climate in the NHS, is to think about the impact of introducing change and how it impacts on the service as a whole. And then I think there's another question for Zosia, which is you mentioned a situation where an embryo or fetus was thought to carry a disease-related gene variant, but it was found that the couple's other children carried the variant and were unaffected. Can Zosia and the other speakers please explain how the concepts of penetrance and expressivity might account for this, and what the difference is between the two concepts? So Zosia, do you want to go first on that? Okay. And then if you want to pass over a bit of the question to Jonathan, perhaps.
I'll try and explain. So first of all, very interesting to hear about the rapid exome testing and the impact on options therefore for future pregnancies coming through. This is something we're not seeing yet in Scotland because our funding framework is different, but it's coming. With respect to, and we welcome it coming because it's opening up options for families. With respect to the question about the penetrance and expression of variants, the specific case I was describing is not about that. And I think the application in most of this sort of variant characterization work area is that in what had happened in that case was that there was a gene difference that was predicted to be disease causing by one, the people reporting it, but actually it didn't cause that disease because the prediction was incorrect. And we work in this world of taking variants and looking at them, and we have a number of ways that we use those to predict whether they'll cause a disease. So the one we love to see is when a gene alteration cuts a protein short, and traditionally we've thought that that's a pretty good hit, particularly if it's the person in front of you is the first person with the disease and it's the right gene for the way they look and so on. But we still find that sometimes a truncating variant isn't necessarily the cause of a disease. So that's you know, but what is more difficult is where you get an alteration of a base that might be in a bit of the gene that's predicted to change, you know, be a really important bit of the gene, but sometimes a substitution of one base for another doesn't change the way the protein works. And it depends where it is, it depends which substitution it is, and Nicola can maybe speak more about this, but basically it's a whole art and science of the work that the clinical scientists do. Is they spend every day looking at sequence variation and deciding if it's disease causing or not, and it's difficult to do, it takes time, and that's why it's just not, this sort of carrier screening side of things has not been prioritized, and it always will leave uncertainties for the foreseeable future. As the databases develop, as world knowledge develops, it will get better and easier. We'll kind of have a truth set, but we haven't got that now. So this isn't, that particular case is not about expression or penetrance, but there are certainly other diseases that we see in the clinic where that is the case, like in tuberous sclerosis families, you can have mildly affected and severely affected people with the same variant. Shall I hand that over?
I think this goes back to knowing what you're testing for, because once you are seeing a child who has a difficulty, they may have a very clear clinical pattern that pattern tells you which gene you should look at, and that's a very strong piece of evidence when you see a variant in that gene that this is causing that disease. But when you're dealing prenatally, you're dealing with an ultrasound scan, which is a slightly fuzzy grey picture of a baby. And sometimes you have hard handles, things that give you really good signs, such as shape of bones and bone growth or head shape. And sometimes you have really soft signs that are telling you there's something worrying, but you're not quite sure what it's about. And then when you get back to sequencing embryos, they are just little balls of cells. And if we sequence every gene and say, well, there's a variant in the tuberous sclerosis gene, or there's a variant in this skeletal gene that causes short limbs, you don't know, or it's much harder to predict what the effect of that variant is. So if a patient already has a condition, you can map it back to the genetic change. If you're finding the genetic change in the absence of a condition, you could be left in a situation where it really makes it much harder to know what is the implication of this variant I have found. It looks like it's disease causing, but really you cannot be as certain because of the lack of clinical mapping. The second situation where this makes a really big difference is when you have genetic variation that causes a very wide spectrum of disease. Say you have a chromosome deletion that causes some people to have quite significant learning difficulties, but other people will develop relatively normally and have normal intelligence for reasons that we don't fully understand. And in that situation, testing for that genetic variant is actually clinically problematic. Oddly, in a pre-implantation genetic testing situation, if I'm looking for one of these variable penitence chromosome things, dad has it, he's fine, dad has a daughter who's quite severely affected, can we do a test in this pregnancy? Pre-implantation genetic testing for aneuploid, for segmental duplication, can be really useful, because then we know that the embryo we reimplant and 50% of the embryos generated will be fine. Actually, that's one of the few situations where the uncertainty can be got around using PGT in the clinical setting. So it's to do with how well you can map your clinical picture to the genetic variation that you're seeing.
Are there people you would advise definitely to have these tests, or others who need not to use them?
In the perinatal setting, this is a core function of clinical genetic services with clinical geneticists and genetic counselors. So whether or not to have a genetic test is, it's never something you would be advised to have or advised not to have, but it's something you'd be given the information about, and you could choose to have a test if it was a valid test. So your best route of referral, discussion, support for your patient is your friendly neighbourhood clinical genetics department, who should be configured to help with that.
I think that also makes me think about where patients who, are fertility patients and are perhaps sometimes being targeted for marketing of some of these tests, who don't have, as far as they're aware, a genetic condition or carry a genetic condition, but are thinking about, slightly worried and thinking, well, where can I look for some reputable advice? They don't want to go to the GP with it, or they wouldn't be able to sort of necessarily access their friendly neighbourhood clinical geneticist about this. But what websites or resources are there out there, do you think, that you would signpost people to who are looking for information?
First of all, I will start with HFEA. It is usually a good place where to start because it lists the clinics that are licensed in the UK. And because they are licensed, obviously, this is where you can have your treatment, but it also means that there has been a process to review how they work. And they also provide quite a lot of information for patients regarding tests available, pathways available, some very general informations, but they also signpost on where you can have an appointment to understand whether this is the right choice for you or not. So this will be the first, I think, point of contact. Obviously, PET might be another option, but probably for lay people, it is more known that there is an organism like HFEA, that specific charity who gives specific information. Sometimes also places like Unique or something like that might also provide good information, but obviously you will only access this route if you have already some relatives with a condition for which you have been signposted to Unique.
If we're talking about having, for example, pre-implantation genetic testing, the time to have that is if you've had a previous child with a condition, then there can be an NHS assessment of whether you'd get funded treatment. And at that assessment, there can be a chat about the pros and cons. It's not an easy process to go through. Not everyone going through it has a baby at the end. It may not be the best option for you, but the genetics department can discuss that if it's about PGT. If you've got fertility questions, I would direct yourself to your fertility clinic. And if you don't have either of those needs to be thinking about using reproductive technologies, then I would think really, really hard about it, because it's probably not the best way.
Karen Sage actually has posted in the Q&A to remind us about Genetic Alliance UK and the useful resources that they will have on their website. So thanks for that, Karen. Francesca implied that the reason polygenic risk scores are increasingly known as polygenic scores is because of doubt about how much they can tell us. But the behavioural geneticist Robert Plomin has been arguing for years that they should simply be called polygenic scores because he thinks that their usefulness goes far beyond medicine and therefore beyond questions of risk. So Francesca, do you want to have first crack at that? And then we'll see if anyone else wants to comment on
I think it's a very important point. So there are two main things that I want to touch. The first is that I did not imply that the scientists tend to use or tend to move to polygenic scores instead of polygenic risk scores because they don't think they are valid, what they do, but because of the risk of misuse of this information out of context, and maybe to be rushed in some contexts before they will be properly assessed in trials, in pilots, and so forth. So they want to move out about the risk thing and remain in the area of association. And I fully agree that this is beyond just disorders, but is also a matter of traits. And this is where a lot of ELSI, ethical, legal, social issues might also come into the discourse, because quite a few of the conditions, let's say, that they are proposed for are actually also individual traits in a way. They're not necessarily disorders. So there is like a continuum, because what we define a condition like a cardiovascular problem or diabetes, what we define neurodiversity, what we find individual trait. So an example here is, this is a very famous case that has been published by a journal recently of one of the first couples, for instance, buying this test for PGTP. And both the prospective mother and prospective father have actually autism, and they have downloaded the data, uploaded to other sites in order to have also a sequencing and other polygenic score for a number of neurodiversity traits, not conditions, in order to select what they thought was the best embryo for their family. They have on purpose not looked into the risk for autism, because for them, autism is positive, is their identity, is something that they are not a problem with.
I feel it's rather controversial trying to identify trait that you would quite like to have in your child rather than ones that are associated with disease processes. And there is scope for a large and actually quite interesting ethical debate about whether it should be permitted, but also as part of healthcare, it's clearly not really within scope to be using information in that way. There are actually pros and cons to whether it should be a permitted thing to happen, but there are many good reasons why it is not part of standard healthcare practice, because it's not really based around a disease process as such. And then we're often down to how predictive, again, these things really are.
And Zosia.
I find it interesting that we hear these stories, and yet what I see day to day in my clinic is couples who've been accessing PGT, and the actual number of viable embryos that are free of the disorder, the highly penetrant disorder they want to deal with, is actually very small. And even at the end of 3 cycles, they may not have one that works. So once you start layering on additional things to select for, I think it gets very, very difficult. But also, we're talking here about sort of continuous traits where you've got natural variation, you've got a huge environmental component, and yes, you're trying to select the genetic stuff and give it the best shout, but there's so much variation that it would, I think there are things that would just be very, very difficult to predict, especially with the state of knowledge we now have. We've all got lots of mutations in our DNA, and only some of which cause us diseases, and we're learning how to work out which ones of those matter, but we're still a long way from finding that out. So I would say it's not the best way to spend your money.
And so I wondered whether you have concerns, or there are potential concerns about sort of parity and equity between people who are fertile and just, don't need any help, and not necessarily therefore accessing services in the same way as people who have a fertility problem. So, perhaps a fertile couple would not necessarily be able to access tests or screens that an infertile couple perhaps may be able to access more easily in an IVF unit.
I think it's largely difficult for me to answer, because within our PGT team, we actually see patients who have a rare disorder, usually, or in any case, a disorder which is genetically determined, which is not the same as considering couples who want to have infertility treatment because they are infertile, but they do not have PGT. So we see a niche, I think, of this patient. In terms of equity, this is definitely something that should be kept in mind for a test like, for instance, the expanded carrier screening. Because as I mentioned before, the couples who are infertile or if they undergo assisted reproductive technologies, they don't really have, with the exception of cystic fibrosis in males, they don't have a significant increased risk comparing to the couple of the general population for autosomal recessive disorder, which is mostly what the expanded carrier testing will be for. So in that case, if we will decide theoretically in the future, this is a good value for money, then there will definitely has to be a discussion regarding equity and offering this to the general population as well, not just to those who access infertility clinics. For other investigation, obviously, it depends on the infertility per se, so it's different.
I have in the clinic had quite a number of discussions with our fertility team where people are coming to fertility treatment, and the history of a genetic disorder comes up. It's not their primary reason for going for fertility treatment, but it's kind of, it's a bit like you're in the frozen foods aisle already. Would you like some meatballs as well as the frozen veg you came for? And we have those discussions, and often if the patients haven't driven that question themselves, once you go into the limitations of PGT and the fact that you then have to discard embryos, often we find they don't follow through on it. But I think it is an increasing question, and it's a point well made. I would also say in our discussion, some of our discussions in Scotland around when perhaps carrier testing should be offered, the conversation has gone, well, why should people with having fertility treatment get something we're not offering to the rest of the population? If we offer this, it should be offered to everybody. And what I think is interesting is, it seems to me, and I might be wrong with this, that the private market in carrier screening is very much driven by the fertility market rather than the general population market. But that's perhaps because it's income selected. I don't know.